Generally, the family of nuclear receptors have a low target affinity, high amino acid sequence homology, and similar DNA response elements. How then are they targeted to their appropriate genes? This slide explains how nuclear receptors binds to the target DNA in a specific way. Here, 3 different heterodimers, RXR and VDR, and RXR and TR, RXR, PAR. You can see the full name of each receptor. RXR actually stands for the nuclear receptor for cis-retinoic acid. VDR, receptor for vitamin D. TR, thyroid hormone receptor, and RAR, receptor for the trans-retinoic acid. Heterodimers are different, but share this RHR in common. You can see the DNA response element for each heterodimer. Here you can see the binding region in the red color has a direct repeat, same direction. Another interesting thing is that the two response elements actually has in the middle with different lengths of the spacer region. For the first one, 3, 4. For the second one, 5. For the third one. In other words, different heterodimers involving the common partner RXR can recognize response elements of identical target sequences but with different lengths of the spacer regions. Third type of zinc containing motif will be binuclear zinc cluster. It has 2 zinc and 6 cysteine. The most thoroughly studied member of this family, GAL4, activate transcription of genes required for the breakdown of the sugars, galactose and maltose. GAL4 binds to DNA as a dimer. GAL4 has 3 domains: dimerization region and DNA binding region, which contains zinc cluster and linker regions between them. Dimerization regions, they make a coiled-coil structure. DNA region is 17 mers. Schematic diagram of DNA binding zinc cluster region of the GAL4 is shown in this slide. In A, zinc cluster contains 2 zinc atoms in the middle, each bound to the 4 cysteine residues, 2 of which bridge the zinc atoms. You can see the amino acid numbers in the loop regions between cysteines. Okay, these regions are variable, however, the others are highly conserved. The B structure shows the actual structure. The red regions correspond to this region in A, provide the sequence-specific DNA interaction with DNA. The zinc cluster stabilize the structure to give proper fold for DNA binding. Here you can see how zinc cluster binds to the DNA. C terminal of first alpha helix, the red region, is being inserted into the major groove of target DNA. You can see the half of dimer with each binding region. In B, you can see the several types of interactions between them. There is only one single specific bindings that occur by lysine 18, and also yellow region explains the binding between protein and bases. But in this case, main chain CO carbonyl group of lysine 17 and 18 just contribute in binding to the edge of bases. There are several non-specific interactions between DNA, phosphate, sugar backbone, and the linker regions in DNA. You can see the blue region and also corresponding binding regions in DNA. In previous slide, you saw massive interactions conferred by linker regions. So zinc cluster or linker, or possibly dimerization element, which region is responsible for specific binding to its target DNA. Domain swapping experiment may give an answer to that. In A, you can see the GAL4, in B, PBR1, which has different binding specificity, GAL4 and PBR1. In C, zinc cluster from the PBR1, but joined with linker and dimerization element from GAL4. This chimeric protein binds to the GAL4, meaning linker and dimerization element is responsible for DNA binding. In D, you can see zinc and linker from the PBR1 joined to dimerization element of GAL4. This chimeric protein binds to the PBR1. Together, link region actually determine the specific binding to the DNA. Next transcription factor will be leucine zipper. The leucine zipper motif was first recognized in the amino acid sequences of several transcription factors, including yeast transcription factor A and CEBP, mammalian transcription factor, and several oncogenes. As you learned in chapter 3, the sequences of this protein are plotted on a helical wheel. The 7 residues called heptad showing some clear the picture, which is highly conserved repeating leucine here and here and here and here. You can see the position of each heptad outside: A, B, C, D, E, F, G. So 7 residues just repeating and align the exact same position on the helix in coiled coil. There are two major interactions in this structure: hydrophobic interactions between A and D, and charge interactions neighboring residues. Leucine zipper could make a heterodimer that could alter their DNA binding specificity. In this schematic drawing, you can see two binding sites by two different homodimers, but their heterodimers could recognize different binds to the hybrid DNA sequence. Next, I'm going to explain how leucine zipper binds to the DNA. GCN4 has a DNA binding domain in C-terminal. You can see C-terminal sequences ending with zipper region, but the preceding regions called basic region because it has a high content of arginine and lysine. You can see the binding the DNA regions, which is basically palindromic structure, but additionally modified DNA, where you can see the one addition of one nucleotide in the middle or two, also could be recognized by this protein. The structure is very simple. Each monomer of GCN4 domain forms a smoothly curved continuous alpha helix comprising both the basic and the leucine zipper regions. Monomers are held together in a dimer in the zipper region. They diverge from each other in basic regions, which are bound to the DNA in the major groove on the opposite side of the basically unchanged B DNA. It's like a helical forceps gripping DNA. This kind of mode could explain, its flexibility for the DNA. Remember, there are one or two nucleotide being inserted in middle. Of course, there are specific and non-specific interactions between them, but I'm going to skip this one. Next category will be HLH, helix loop helix. You can see three different this category aligned together. All together have basic regions and followed by zipper or HLH or two of them. Basic regions are responsible for DNA binding, the other for the dimerization. This is from the MyoD transcription factor. You can see the helix loop helix region. Two helices actually position almost parallel. Basic regions just connected smoothly again to the first alpha. How they interact? Actually, they make a dimer forming four helix bundle with the loops being positioned outside of a bundle. This shows how they interact. It's very similar to the basic leucine zipper. The basic regions involved in the DNA binding. Again, explaining the specific interaction and non-specific interaction between them here. And the next group will be BHLH zip, having two domains. The single monomer has this kind of smoothly connected zipper, alpha helix to the HLH, followed by basic regions. How they interact is very similar to the previous one. The aligned sequence shows some common things in two categories: hydrophobic
holes and one lysine, but buried in loop regions.